Good afternoon, guys. Welcome to today's session. My name is Belinda, and I'll be presenting today's session. Before we start, please on and your speaker volume is turned up. Please also note that you are automatically muted when joining the session on the handout section. Okay, you are encouraged to ask questions and leave comments. However, please be sure to leave comments that are relevant to today's content. If we did not get to your questions, please send an email to academic at impact.co.za. Visit the impact. So let's just give it a few minutes, a minute or two. For everyone who's here, by an indication of a hand, can you hear me? Please indicate by raising your hands if you can hear me, please. Thank you. Thank you. Okay, we're just gonna give it a couple more minutes just to give people some time to get ready, and while other attendees are joining in. You are encouraged to ask questions where you don't understand. To improve the quality of my connection, I have switched off the webcam. Okay, so that I make it possible for you guys to hear me. Okay, otherwise with my webcam on, my voice is going to break up a lot. Okay, so today we'll be looking at genetics, the study of inheritance from parents to offspring. In the previous webinar, we've looked at a monk, an Austrian monk known as Mendel, who was known as the father of genetics. And we've learned that through his studies or through his experiments, or while experimenting with pea plants, he discovered that there was a certain elementin that was passed from a parent to an offspring. Okay, and it was later discovered that that elementin was known as genes. Okay, so genetics is the study of heredity. So basically, we're going to be looking at how certain traits are passed on from parents to their generations. Okay, to start off, we've learned that, or we know that the human cell contains 46 chromosomes altogether. Okay, and this chromosome contain the DNA that makes each cell unique. Okay, this is a human karyotype, a typical human karyotype. And as you can see, we've got 46 chromosomes altogether, okay, or 23 pairs. 1, 2, 3, 4, all the way up until pair number 23. The last pair, pair number 23, is known as the sex cell. Okay, so this determines if whether a baby is going to be a female or a male. Okay, we know that if a human is a female, that last chromosome is going to be an XX. It's going to be 2 X's. Okay, or if we are looking at it at this way, if it's a female, both these chromosomes are going to be more or less the same size. The chromosomes are going to be more or less the same size, but for a male, the last chromosome is either going to be denoted as XY, or one of the chromosome is going to be shorter than the other one. Okay, so this means that this cell is a cell of is it a male or a female? Can you indicate to me on your question box if this is a male or a female? Remember, we observe the last pair, chromosome pair. Okay, it is a male if the one chromosome is shorter than the other one. It is a female if the chromosomes are the same size, like you see here. All the chromosomes are more or less the same size. Okay, so observe the last chromosome. Is this a male or a female? Okay, well done. It is a male, because as you can see in that last chromosome, that one is much shorter than that one. Okay, so this is one way in which a karyotype can be presented, or they can use X's. So with that being said, that last one will be an XY for a male and an XX for a female. Okay, from this karyotype, one of the chromosome you inherited from your dad and the other you inherited from your mom. Together, this pair of chromosomes give you your whole cell, or it gives you your whole genetic makeup. So for each pair, one you have inherited from your mom and the other one you have inherited from your dad. Okay, so you got 23 chromosomes from your mom, which was made possible by the process of meiosis. So that cell division where the number of chromosomes are halved inside the sex cells of the father and the mom. Okay, so meiosis made it possible for the sex cells to contain only 23 chromosomes instead of 46 chromosomes. Okay, so that during fertilization, the 23 that your dad gives you and the 23 that your mom gives you together will give you 46 chromosomes that will make you a complete human. Okay, all right. Let's move on. So these are the terminology that are used in genetics, and for you to be able to understand genetics properly, or for you to see that this is actually an easy topic, first thing that you need to do is you need to understand what the terminology is. What are they talking about when they say allele? What is dominant? What is recessive? What is a genotype and what is a phenotype? What is a parent and what is an offspring? And what is a gamete? I mean a gamete. Most of this terminology you already know. Okay, so now you just need to affirm it and you just need to understand it. Okay, and yeah. Okay, so all characteristics are controlled by a minimum of 2 genes. Okay, all characteristics are controlled by a minimum of 2 genes, one of which you inherit from your mom and one of which you inherit from your father. These genes give the characteristic a different of presenting itself. Okay? Different genes that control the same feature are called alleles, and we're gonna look at this in the next couple of slides. What alleles are by looking at a picture. Different genes that control the same feature are called alleles. Okay. Now let's look at genotype and phenotype. Genotype is the letter or term used to describe the allele of an individual gene or pair of genes. A genotype is the letter, we use letters to represent genotypes, is the letter that is used to describe the allele of an individual gene or pair of genes. Okay, it can be a gene for eye color or a gene for a hair texture, or it can be a gene for face shape, it can be a gene for the color of a plant, it can be a gene for the length of a plant. So a genotype is a letter that represents an allele for an individual gene or a pair of genes. Okay. A phenotype is how the gene shows itself or how it appears. So what we observe from the outside is called a phenotype. The genotype is the representation of the genetic makeup using letters. The phenotype is what we observe from the outside. Okay, so it is how the gene shows itself from the outside. Okay, we're going to look at examples in the next slide. Dominance and recessiveness. Some alleles for a feature are able to mask the influence of the weaker gene when they are paired. So during fertilization, when the allele from the male combines with the allele from the female, some alleles have a feature, or some alleles are able to mask the influence of the other one. Okay, the alleles that are able to mask the influence of another allele are known as the dominant allele. Okay, this does not go by sex. It does not go by whether it comes from a female or a male, that a male is stronger than a female. It does not work like that. Okay, sometimes an allele from a female will dominate an allele from a male. Okay, so you'll get, your eyes will look much more like your mother's than your father's, because your mom's allele was dominant over your father's allele, which was weaker. So it's got nothing to do with their sex, with whether you're a female or a male. but it's all got to do with the genes. Okay, dating back from from ancestors or from previous generations. Okay, some alleles are much stronger than other alleles. Okay, the stronger gene is dominant and is represented with a capital letter. The stronger allele, or the one that masks the weaker one, is known as the dominant allele, and it is represented with a capital letter. Remember, we said a genotype is a representation of a gene using letters. Okay, so this would be a genotype of a dominant allele or a dominant gene. The masked gene, the one that does not appear in the phenotype, the one that is weak, or the weaker gene, is known as recessive. Recessive means it is hidden. It is hidden. It was completely masked by the dominant allele. Okay? You see this in friendships as well. Some of our friends are much more dominant than us. Okay? So even when we are outside playing or doing whatever, because they've got dominant characters, most of the time you will be hidden. Most of the time that the weaker ones are not even recognized or seen. Only the dominant ones are popular. The much more weaker ones, or let me say, yeah, the weaker ones or the less dominant ones are not that much popular. They are not observed or seen by people. So same goes for genes. Some genes are recessive. They are weaker and hence are masked by the dominant allele. Okay, we used a lowercase letter to represent recessive genes. Okay, but for each characteristic we use the same letter. Okay, so if we are speaking of color, eye color, let's say your mom's color is brown, your father's is black and recessive. Okay, if we were to represent the genotype of the offspring, okay, let's say you, the offspring, have brown eyes. Okay, this means your mom's eye color was dominant over your father's eye color. Okay, although these are different colors, this is brown, this is black. What I'm trying to say is we use the same letter type to represent them because they represent the same trait. They are all traits for eye color. They are all traits for eye color. So we're going to use the same letters to represent them. Although we will represent the dominant one with a capital letter and the recessive one with the small letter. So just because it's different colors doesn't mean we'll have to use different letters to represent them. Okay, we use the same letter to represent them because they represent the same trait, they represent the same allele, and that is the allele for eye color, right? So we spoke of an allele in the beginning, and we said different genes that control the same feature are called alleles. Okay, so let's see them on a chromosome. So we've got different genes here, one from a female and another from a male. Okay, this is the gene for eye color, I mean for the petal color. We're speaking of plants now. These alleles are found in the same place in the chromosome. Do you see that they match each other? Although one is from a female and one is from a male. Okay, so these different alleles found in the same place that represent the same trait are known as alleles. And this, in this example, we have the allele for petal color. Okay. Yeah. Okay. Now let's look at homozygous and heterozygous. Okay. Remember, homo means the same. Zygous means that comes from the word zygote. Okay, so homo means the same. If the alleles in a matching pair are the same, they are called homozygous alleles. If the alleles in a matching pair are the same, they are called alleles. They are called homozygous allele. Okay. For example, this is from a male. This is from a female. But then they are all alleles for yellow petals. They are exactly the same, or they are both dominant, or they were both recessive, but they were exactly the same. So we refer to those as homozygous allele. Okay. These were also alleles for red petals. We found exactly the same alleles from both parents. Okay, so this is known as homozygous alleles. If the alleles in matching parents are different, they are called heterozygous alleles. Again, hetero means different. Okay, for example, from the male, let's say we got an allele for red petals. From the female, we got an allele for yellow petals. So because these were for the same trait, which is petal color, but because it was different colors from the different parents, we call this heterozygous alleles. Okay, so this means in the genotype, one will be represented with a capital letter and one will be represented with a small letter. Okay, so this means either one is going to be dominant and the other one will be recessive. Unlike with homozygous, either both of them will be dominant or both of them will be recessive. So either they will be represented with the same capital letters or the same lowercase letters. Here they will be represented with, one will be represented with a capital letter and one will be represented with a small letter. Okay, if you are confused, just hold on, it will all come together. Okay, or you can just send me a question on the question box. Okay, if you were lost somewhere. Okay. Some alleles are dominant to other forms of a gene and will be expressed in the phenotype. So the dominant one will be the one that is expressed in the phenotype. It will be the one that we observe when we are looking at you or when we are looking at the plant or whatever we are studying. The dominant, the allele with the dominant, the dominant allele will be the one that's visible to us. Okay. So we have established that letters are used to represent different alleles. A dominant allele is always a capital letter. For example, this is an allele for red petals. We will use capital letter R. Okay, and because we've used the capital letter R, we know that it's dominant. A recessive is always the corresponding letter, but lowercase. So if we were going to look at an allele for yellow petals, we're going to use the same letter, okay, because they're representing the same trait. But because this one is recessive, we're going to use a lowercase letter R, okay? We're going to see what happens if both of them are dominant. We're going to look at that later, okay? The allele pair of each characteristic is called the genotype. So when we pair the two together, we will have our genotype. So this means the color or the genotype for this flower would be RR. This is what the genotype of this flower will be. If we were to pair them together, this is what the genotype would be. Okay? R, big letter R standing for red and dominant. Small letter R standing for yellow and recessive. Okay? Remember, the dominant allele is the one that we observe in the phenotype. It's the one that we observe from the outside. So what do you think the phenotype of this flower will be? Can you type it in your question box, please? What do you think the phenotype of this flower will be? What will we observe? Will we see a yellow flower, or will we see a red flower? What will we observe in the phenotype? Excellent. We will observe red. So the flower will be red from the outside. Why? Because the red is dominant over the yellow. We see it with the capital letter. Okay, we see it with the capital letter. Okay, so this means our phenotype will be red flower. Okay, now we're going to be looking at genetic crosses next. So during a cross, we can observe one of three scenarios. Okay, sometimes we can get complete dominance, sometimes incomplete dominance, and sometimes codominance. Okay, complete dominance is when a dominant allele completely suppresses the appearance of the recessive allele. Complete dominance is when the dominant allele completely suppresses the appearance of the recessive allele in such a way that from the outside we only see the dominant allele. So if you were to look at the plant, you wouldn't even tell that there was that trait, the recessive trait, just like with the previous example. When we paired these two, although this plant has genes for yellow, this completely suppressed it. So this means all we observed was a red plant, although it's got genes for yellow. Okay, so it's complete dominance when the dominant allele completely masks the appearance of the recessive allele. Okay, incomplete dominance means there are no dominant and no recessive alleles. The offspring will show a blend mixture of the trait, a blend or a mixture of the trait. So none of the genes from the parents were dominant or recessive, but together they formed a completely new blend of the trait. For example, let's say we were mixing or breeding a white and a red plant, and after breeding the result was pink. Okay, so neither the red nor the white was shown in the phenotype, but a mixture of the two, which was pink, was shown in the phenotype. This is known as incomplete dominance. So we have no dominant or recessive alleles, but the offspring shows a mixture of the two. Okay. Co-dominance, both alleles of the trait are equally dominant and both will show in the phenotype. So in this case, both alleles of the trait are equally dominant and both will show in the phenotype. For example, if you get they call it an Oreo dog, okay, it had a mom who, let me use proper colors. Let's say the mom was black, had a black coat, and the father had a red coat. Okay, they breeded and they formed, or they bred and they formed a puppy that had both red and black spots in its coat. Okay, so this pup had traces of black from the mom that it inherited from the mom, and traces of red that it inherited from the dad. So both of them were dominant and both of them show in the phenotype. Okay, so this is known as codominance. They co-dominate. Both of them dominate together. So both of them will be expressed in the phenotype. Okay. Lastly, let's look at another term that we're gonna find a lot in genetics. It's called sex-linked inheritance. This simply means the allele is carried on the X chromosome of the genome. Okay, of the that very last chromosome pair in the karyotype. This allele, sex-linked inheritance means this allele is carried on this chromosome that is found in the very last chromosome pair in your karyotype. Okay, right. Remember, we use Punnett squares in order for us to solve monohybrid genetic problems. We use a Punnett square in order to solve genetic problems or monohybrid genetic problems. We're going to be looking at dihybrid. If we still have time, we're going to be looking at dihybrid genetic problems as well. Okay, so you need to know your template very well. And if you use this template throughout your questions, you shouldn't have a problem. You shouldn't have a problem with this. You should find it very easy. Can you guys still hear me? Please raise your hands if you can still hear me. Thank you. Sorry, I'm going to constantly check just so that I don't lose you guys on the way. Okay. So in our template, the first thing that we need to do is we need to look at the phenotype. Okay. By phenotype is how do both parents appear? Okay, depending on the trait that we are looking at. If it's eye color, what is the eye color of both? Is it blue and brown, or is it green and blue, or what? So first we look at the phenotype, the appearance of both parents. Then we look at the genotype, the two alleles of both parents. In your question, they will tell you which one was dominant over which one. They will tell you which one was dominant and which one was recessive. They will also tell you if they were homozygous or heterozygous. So you just need to read your question carefully so that you can gather this information. Okay. Then after meiosis, remember meiosis is that reduction division where gametes are formed. Gametes are your sex cells. Okay, gametes will be formed, and these gametes during fertilization are going to fuse together to form our offspring. Okay, so from our genotype, we're gonna get our gametes. The gametes from the female and the gametes from the male. It does not matter where you put what. Okay, from there we combine the gametes in our Punnett square. I will show you with an example now to check or to get what the possible genotypes of the offspring will be. Okay, from there, by looking at the possible genotypes we got from our Punnett square, we will write our phenotype. Okay, you can either use this format or you can simply go to the Punnett square. But list the appearance of both parents, list the 2 alleles of both parents. From there, go straight to your Punnett square. So you can use either one of these two formats. Okay, a typical genetic question will give you about 6 marks. It counts about 6 marks. Okay, so it's very important that you guys understand this, because 6 marks is a lot to lose, right? So let's look at a question, an example. In humans, the ability to roll the tongue is due to a dominant allele. The ability to roll the tongue, so this is the trait that we're going to be looking at. The ability to roll a tongue is due to a dominant allele. Okay? A man who is heterozygous for tongue rolling, we already got information. This man is heterozygous. So this means his genotype is going to consist of a capital letter and a small letter. Okay, remember heterozygous means different. So this means in his genotype, 1 letter is going to be a capital letter and 1 letter is going to be a small letter. Okay, a man is heterozygous for tongue rolling, but although this man has a heterozygous a genotype, he can roll a tongue. So this means he's got the dominant allele for tongue rolling. He has it. Okay, and a woman who cannot roll her tongue have children. So we have a man who is heterozygous for tongue rolling and a woman who cannot roll her tongue. They have children. Use the symbol T and small letter t for the alleles of the tongue rolling characteristics and represent a genetic cross to determine the possible genotype and phenotypes of the children. Remember, they don't say what will the genotype of the phenotype, the possible genotypes. Okay? And the question is 6 marks. I'm going to read the question again. A man who is heterozygous for tongue rolling and a woman who cannot roll her tongue have children. Use the symbol T and T, big letter T and small letter t for the alleles of the tongue rolling characteristic and represent a genetic cross to determine the possible genotype and phenotype of the children. What must you look for first? First, we need to look at the characteristics. What are they? For the male and the female. What are their phenotypes? Okay, the male can roll a tongue. The female cannot. Non-tongue roller. Okay, those are the characteristics that they gave us here. The male can roll a tongue and the female cannot. What do we do next? Next, you need to identify the dominant and recessive trait. The dominant is tongue rolling and is represented with a capital letter T. Tongue rolling is dominant, and then recessive is non-tongue roller and is represented with a small letter t. How do we know that? They told us here. In humans, the ability to roll the tongue is due to a dominant allele. Okay. Number 3, we need to determine the genotypes. Okay. Is the person homozygous or heterozygous for the trait? Let's start with the father. The father was heterozygous, so this means that he's got different letters. One will be a capital letter T and the one will be a small letter t. We must have that capital letter T because he's a tongue roller. He's got this dominant allele. Okay. This, the female is a non-tongue roller, meaning he's got the recessive allele and it's homozygous. Okay. If she had capital letter T as well, it will mean that she can roll a tongue. Okay, but they told us she's a non-tongue roller, so this means she will have recessive genes of this tongue rolling characteristics, small letter T. Okay, so now that we've determined the genotypes, we can go on into our Punnett square. Okay, so tongue roller is dominant T. That means that non-roller will be recessive small letter T. To be a roller, it must both be capital letters, both dominant alleles, or you must at least have one capital letter. Okay, let's look at the solution. Let's look at the phenotype. First, we had a tongue roller breeding with a non-tongue roller, step 1. Step 2, we determined the genotypes. Because it was heterozygous, one will be a capital letter and one will be a small letter. Okay, this one could not roll a tongue at all, so this means he'll get the recessive genes. From there, meiosis takes place and we'll get our gametes. One will be big letter T and the one will be a small letter T. Here we're gonna get two gametes that are the same, both small letter T's. Next, fertilization. Go to our Punnett square. Put our gametes. Start with the male. It does not matter. You could have put the male this side as well. Does not matter where you put them. Okay, so we put our male there. Let me just do it myself. Then we're gonna have our female there. Next, we cross them. So here we're gonna have capital letter T and small letter T. Here we have T and T, small letter T's. Here we've got big letter T. It's always better to write the capital letter first. Big letter T and small letter T. Here we've got big letter T and small letter T. These are the possible genotypes that the offspring will get. Okay, the offspring will either get that phenotype or that phenotype or that phenotype or that phenotype. So if an offspring comes with this genotype, both capital letter T's, it means something is wrong here. Okay, so we're gonna think that the kids were switched in the hospital, because by looking at this Punnett square, it does not show a possibility of ever getting this type of genotype. So if we do our Punnett square, or if during some test we observe this genotype in one of their offspring, it means that something is wrong. It means this is not their baby. This is not their offspring, because these are the only possible genotypes that their offspring will have. Okay, let me erase that. Okay, so by looking at this, 2 out of the 4 will be heterozygous for tongue rolling, and 2 out of the 4, or 50%, will be recessive, homozygous recessive for tongue rolling. Okay, 50% of the offspring, or 50% of the possible genotypes will be, or 50% of the possible phenotypes will be heterozygous, dominant for tongue rolling, and 50% will be recessive, homozygous recessive for tongue rolling. It's called homozygous because both of them are small letters. Okay, homozygous recessive because both of them have a recessive gene. By looking at that, we see that by looking at the fact that they are represented with small letters and not capital letters. Do you guys have any questions? Do you guys have any questions? If you've got questions, please type them on the question box. Can you do a square where we look at the grandchildren with eye colors? You can do the square from one generation to the next generation. And yes, you can look at eye colors as well. You can do the square to look at all sorts of traits. Eye colors, the texture of the hair. Will the grandchildren have curly hair or straight hair? Will the grandchildren have blue eyes or green eyes? Or you can go from one generation to another. No, it will not, the square will not look different. It will be the same. Okay, but the genotypes of the offspring can be different sometimes. Okay. Okay. What happens with incomplete dominance and codominance? Okay, what do we represent? What do we use to represent the genotypes then? Because both of them are dominant. Okay, with incomplete dominance and codominance, we represent the genotypes with capital letters only and not small letters, because both of them are dominant, but we will use different letters to represent the same trait. Okay, so this means if we were looking at a red plant with a white plant, then we would use both capital letters but with different letters. Okay, we only do this with incomplete dominance and codominance. We use capital letters only to represent the genotypes. Okay, let's look at incomplete dominance. Sometimes we don't have dominant or recessive alleles. Okay, so none of the alleles are dominant over the other. This means that the other one will not be recessive as well. The offspring will exhibit a blend of characteristics in the phenotype. Okay, so we're going to use different capital letters. For example, red to represent the red, and we're going to use white to represent the white. Okay, so unlike complete dominance, where we used the same letter but with different cases to represent the different alleles, here we use different letters, but they must both be capital letters. Okay, so this means this red plant was homozygous dominant for red. It had both capital letters representing the red allele. And this plant had both capital letters W representing white allele. Okay, because the offspring is a blend of the two. Or let's do this with a Punnett square. Okay, let's draw a Punnett square to see how this happened. So let's put our gametes there. Okay, so we have the one plant and then we have the other plant. Okay, so combining these two gives us an R and a W. These two give us an R and a W. These two give us an R and a W. This one, R and W. So as you can see, the only possible genotype for the offspring will be RW, which represents a complete blend or a blend of the two, in this case being pink. Okay, so the plant is not gonna exhibit any of their parents' color, but a blend of the two, a new color which was formed by a combination of the two genes. Okay, again, if we were conducting a genetic cross and we found a plant that had this genotype, it's obvious and it's clear that this was not an offspring of these two plants. Okay, because as we can see, the only possible genotype that we can get from these two plants is an RW, so a combination of the two, right? Okay, if you've got questions, please send them through. Codominance, both the alleles can be expressed. Okay, for example, red cows crossed with white bull will generate roan calves. Okay, so both the alleles of both parents are represented in the phenotype. As you can see, this calf has some red coating and some white coating as well. Okay, this plant also, this means that breeding was done between a purple flower and a white flower, but because they were both dominant, both these alleles are represented in the phenotype. You can see the traits of both the plants in the offspring. Okay, this is known as codominance. Okay, sex-linked inheritance. The alleles for hemophilia, okay, are indicated as a superscript on the sex chromosomes, on the genosomes. That last chromosome that we talked about in the beginning, that last pair of chromosome in the beginning, the allele for hemophilia are indicated as superscript on the sex chromosome. Okay, so a superscript means it is a value on top of another value. Okay, a subscript you would find at the bottom, a superscript you find on top. So that's what superscript means. Okay, for example, if in your phenotype you get this genotype with capital letter H on top of the axis, this is an indication of a normal female. Okay, again, both, with one capital letter H and one small letter h, this means that although this person has genes for hemophilia, they are recessive. They will not be observed. Okay, so also this is an indication of a normal female. But if you've got small letters, you've got subscript of small letter h, okay, this represents a female with hemophilia. Okay, what will happen for a male then? Remember, the sex chromosomes of a male are an XY. So because this is a sex-linked inheritance, we know that a sex-linked inheritance is only carried in the X chromosome. Okay, so this means a normal male will have a capital letter H in the subscript, in the superscript, and a male with hemophilia will have a small letter H. So a male does not have the luxury of having this heterozygous allele like a female, because they only have one X chromosome in their cells. They've only got one X chromosome in their cell. Okay, we still have a bit of time. The alleles for hemophilia are indicated as superscripts on the sex chromosome. Okay, this is a normal male, this is a male with hemophilia. We've looked at the possible genotypes of females with hemophilia. Okay, so females with hemophilia will have small letter h in their superscript. Okay. What is hemophilia? Hemophilia is a sex-linked hereditary disease that occurs as a result of a recessive allele on the X chromosome. Recessive means we have a small or a lowercase h in the X chromosome. Okay. A normal father and a heterozygous normal mother have children. Represent a genetic cross to determine the possible genotypes and phenotypes of their children. We have a normal father and a heterozygous normal mother. They have children. Represent a genetic cross to determine the possible genotypes. I'm going to let you guys do this one. But first, let's look at the characteristics. And let's try and determine the genotype. So we have a normal father. So this is a father without hemophilia. Breeding with a normal mother, but it's not a normal mother, a heterozygous normal mother, with a heterozygous normal mother. Okay, now let's try and determine the genotypes. Okay, this is the phenotypes. Now of the parents. Now let's try and determine the possible genotypes. Okay, so remember, for a father, it's simple. A male has an XY sex chromosome. A male with hemophilia will have a recessive allele for hemophilia represented with a small letter H. A male without hemophilia will have a capital letter H on their X chromosome. Okay, so this one is simple. This father is normal, doesn't have hemophilia, so this means the X will have an H, a capital letter H in the X chromosome. Okay, this mom is normal, okay, but she is heterozygous normal. This means that, remember, a female has an XX chromosome. Because it's heterozygous, it means we're going to have one capital letter H and one small letter h. Okay? The female will only have hemophilia if she's got homozygous recessive alleles for hemophilia. Okay, this one is normal. Okay, so this means the gene for hemophilia will be recessive. And this definition or this description gives it away because they tell us that it's heterozygous. So this means one will be a capital letter and one will be a small letter. If these two people have children, can you determine the possible genotypes and phenotypes of their children? Can you guys quickly do it in your books and tell me what the possible genotype, or just tell me what the possible phenotypes of the children will be? Okay, we have about 1 minute left, so I will quickly do it. Okay, if we had time, I would let you guys do it. Okay, for those of you who did it quickly, you can give me your answers. You can send them on the question box while I do it here. So the genotype of the father is normal male, mom heterozygous normal. Okay, we're gonna use our Punnett square again. If you've got your answers, please send them on the question box. Okay. Okay, let's put our male there on top. And then let's put our female on the left. Okay, so then for this, we're gonna get a capital letter H and another capital letter H. Okay, these two combined will give us a capital letter H with a Y. These two combined will give us a capital letter H. This will give us a small letter H. These two combined will give us a small letter H and a Y. Okay, so this means there is a possibility. These are the possible genotypes that we can get in our offspring. Normal, female, normal, male, heterozygous or homozygous normal female, or a heterozygous normal female, or a male with hemophilia. So if the question asks, is there a possibility that the offspring of this couple, is there a possibility that the offspring of this couple get a hemophilia? Yes, there is a possibility. Okay, there's a 25% chance that the offspring of this couple, the male offspring of this couple, get hemophilia. As for the females, there is no possibility for the females in this family to get hemophilia. As you can see, all of them are normal. Okay, so this is a normal female, normal male, normal female, normal male, normal female, and male with hemophilia. Okay, let me quickly look at your answers and we can end the session. Please continue going through the slides for dihybrid crosses. They are self-explanatory. They explain this very nicely, so you should be able to process the information here. There will be another genetics classes. Another genetic class. The information will be communicated to you. Okay, so look out for that. Okay, let's look at your answers. Okay, well done. Correct. You guys have given me correct answers. Thank you for joining me today. Enjoy the rest of your afternoon.